Absolutely. Yeah, it's my pleasure to introduce Albert Liu. Albert came to us from Caltech, where he got a bachelor's degree in chemical engineering, and has had an outstanding academic career here in the group. He's won a number of awards for his scholarship. And actually, I'm going to, instead of reading all of these, I'm actually going to cut and paste them in the chat. You can see he's won a number of awards, from poster sessions to graduate student awards. He's been widely recognized by both the AICHE, but also IEEE, and for aspects of his work from the inorganic materials portion of the society, polymers, carbon nanomaterials, and material science. So you get a sense of the interdisciplinary nature of his work, and you'll see that from his thesis. Albert also has shown excellence in teaching, and in fact, and has gone above and beyond what a graduate student would normally experience and participate in terms of teaching here at the institute. I will also cut and paste his numerous teaching awards and recognition on campus here, and this is a Zoom innovation here that we don't have to read through all of these. We can actually put them in the chat. And Albert's extraordinary performance in teaching and research, it's probably not a surprise that he actually has a faculty position in the chemical engineering department at the University of Michigan, and he plans to start there in summer 2022, and he'll be joining Steve Chu's lab at Stanford in a few weeks, where he'll do his postdoctoral work. So with that, you'll see in my own lab, it's been a pleasure to work with Albert. He has pioneered this area of colloidal electronics. He's contributed to energy harvesting mechanisms, both experimental and also theoretical. And so without further ado, I'm going to open the floor to Albert, and he'll present his thesis topic and research to us.
All right. Okay. So thank you, Michael, for that introduction. It's an immense pleasure for me to be here today and talk to you about my thesis research that is around this topic of colloidal electronics. But what is colloidal electronics? It's obviously composed of two words, colloids and electronics. A colloidal suspension can be broadly defined as microscopically dispersed particles suspended within another medium. Now, most commonly, you can find these in liquids or air and even solid. Colloidal suspensions are also ubiquitous in nature. For example, inside the ocean, we have these microscopic phytoplanktons that are swimming around in the solution. The defining feature of a colloidal suspension is this characteristic mobility, because the colloidal particles can travel freely with the solution that carries them. I think electronics bears no further need for introduction, because electronic systems has penetrated into every single aspect of our lives. So bringing electronics onto a colloidal form factor, then simultaneously let us gain the modularity and programmability of an electronic system, as well as the mobility that comes seemingly for free with a colloidal suspension. This opens up possibilities for us to extend artificial electronic materials into previously inaccessible locations, such as the human body for medical purposes, or environmental sensing, and even for reaction design. To put everything in perspective, here's where our everyday electronic instruments lie, at tens of centimeters in scale. Over the past decade, the size of electronic systems has been shrunk to centimeter scale, for example, in these ingestible electronic systems, or even in millimeter scale for smart dust applications. Of course, I think the holy grail of the field is to eventually bring electronic artificial systems into the human circulatory system, or in the inner vasculature, which requires not only for the colloidal electronic particles to be soft and flexible, but also below the size of a human red blood cell, which is roughly around 10 microns in its critical length. So this is the size scale we'll be focusing on our discussion today with respect to colloidal electronics. Now, hand in hand with that small length scale brings a lot of challenges to colloidal electronic research. Solving these challenges has basically formed the roadmap that has defined my PhD thesis for the past 6 years. First, we'll be discussing alternative energy harvesting mechanisms that will bring more energy into colloidal electronic systems. As you probably have imagined, volumetric energy storage, such as lithium-ion battery, does not scale well towards microscopic regime. Well, the only remaining really effective method to power microscopic electronic systems are photovoltaics or solar cells. But as you can probably appreciate, for many of these applications with colloidal electronic systems, we don't really have access to light, for example, inside the human body or inside the crevices of the earth. So it becomes really important for us to be able to achieve alternative ways in which we can grab energy from our environment. In a specific case, we're trying to obtain energy from the solvent environment, as you will hear about in a little bit. The second challenge associated with colloidal electronic system is the ability to communicate between these two particles. When you build electronic systems at the biological cellular scale, electromagnetic wave propagation in the cellular scale is actually very, very limited. We alternatively look at ways in which we can turn that onboard generated electricity into useful chemical signal for signal transduction. Again, learning something from biology. Once we generate that chemical signal, though, in order to complete that communication loop, we have to be able to record and pick up that signal and process it into some sort of digital form factor. And that becomes the third topic. Last but not least, we'll be talking about how we think about integrating colloidal electronic devices into a whole system and explore the collective behavior between these individuals to achieve systems level function that no one individual can achieve by themselves. Let's get started with the energy harvesting. I have highlighted the difficulties of artificial electronic systems to grab energy at the microscopic scale, although this is a solved problem for biology. For example, in these phytoplanktons that are responsible for this beautiful blue color that you see, ATP, which is the biological energy cargo, is transformed into a photon of energy. So how do biology generate their energy cargo? The synthesis of ATP is relying upon the gradient of chemical potential of proton across a cellular membrane. So even though our electronic system cannot utilize ATP because ATP is not our energy cargo, electron is our electrical energy cargo, but we can learn nevertheless something important here, that chemical potential gradient can be used to drive energy production. So are there any examples in biology where we produce electricity? The answer is yes. In our neurons, we fire electrical potentials all the time, known otherwise as action potential. Action potential was generated as a result, again, of a chemical potential gradient of electrical carriers that are asymmetric across a cellular membrane in the form of sodium and potassium cation. So here comes the question. Can we learn this lesson from biology and borrow the fact that you can generate usable electricity from a direct chemical potential gradient of electrical carriers and apply to artificial materials? So first and foremost, we have to select the material. The material of choice here is single-walled carbon nanotubes. So for those of you who are not familiar with carbon nanotubes, you can roughly think of them as rolled up sheets of graphene. And graphene themselves are, again, two-dimensional atomic layers of carbon. So the interesting thing about carbon nanotube is that depending on how you roll up your sheets of graphene, you would get different electrical properties and different electric band structure. And that will correspond to different optoelectrical property of the carbon nanotube. So it's this idea we can very carefully tune the electronic structure and band gap of carbon nanotube with different chirality renders this material very, very powerful. At the same time, carbon nanotube can couple its electronic state very intimately into its solution environment through a process called chemical doping. Just as a brief review of what chemical doping actually is. Imagine that you're in this pool of carbon, which has 4 valence electrons. If I put in adjacent to this carbon lattice an atom or a molecule that is rich in electron, increasing the overall energy of the electron within the carbon lattice. And this overall energy is called Fermi energy. This process is called N-doping. On the other hand, if I put adjacent to this carbon lattice a molecule or an atom that is deficient in its free electron, then the overall average energy of electrical carriers of the carbon lattice will drop in a process called p-doping. Now, if we have learned anything from the biological examples before, we know that a chemical potential gradient of electrical carriers, such as what you see here, can be used to drive a flow of electricity. And it is this very guiding principle that has rendered two of the recent discoveries within the Strano lab. One is called a thermal power wave. The other is called asymmetric chemical doping. Both are for energy harvesting at the microscopic scale. So given the time limitation today, I only can focus on one, which is asymmetric chemical doping, that is more related for colloidal electronics anyway. Asymmetric chemical doping, as its name suggests, is the spatially asymmetric doping event rendered by the solvent molecule. If you can place the lowest unoccupied molecular orbital or LUMO level of your solvent molecule that is close to the valence band of your electronic donor, the carbon nanotube, it's possible for electrons to transfer from the donor carbon nanotube to the receiver solvent molecule. In this case, it's acetylnitrile. In doing so, we can create, again, an imbalance of the chemical potential gradient across the network of the carbon nanotube, and that will induce a flow of electricity. So experimentally, this is very easily done. As you can see, you can just pipette a droplet of acetylnitrile or other solvent doping molecule asymmetrically to the lattice of carbon. And as a result, you would produce a positive or negative voltage depending on where of the electrode you drop a chemical dopant. Now, we're obviously very excited about this phenomena, and we went on to study its exact electricity generation mechanism. Perhaps one of the simplest mechanisms that one can propose is this one. You have your acceptor molecule, acetylnitrile, that absorbs onto the surface of carbon, and then followed by an electron transfer event. The question we want to ask here is whether the first absorption or transport step is rate limiting, or the secondary electron transfer step is rate limiting. To answer that question, we tested 27 other small organic molecules for overall electron transfer rate. In these organic solvents, we're varying in between alcohol, nitrile, and ketone, changing its functional group to tune its electronic structure and electron withdrawing power, and also its steric hindrance for tuning molecular size. The experiment is very simple. What we're doing is we measure the voltage output of all 27 small organic molecules that are electronic acceptors, and we check its initial voltage ramp-up rate. So this dV dt, the slope, differentiates all of these molecules from each other. If you plot this dV dt slope as a function of molecular size, what we see is that there's absolutely no trend in between all of these 27 molecules. So this is a good indicator that this process is potentially not transport controlled. So then we ask the question whether this process could be electron transfer controlled. We plot the same data of the voltage build-up slope as a function not of the molecular size, but in turn as a function of the solvent LUMO level. So to help you understand how to read this plot, the same data you saw from previous slide actually collapsed into a volcano plot. So in this case, when we're stepping from right to left, we're gradually lowering the solvent LUMO level, increasing the thermodynamic driving force of this electron transfer. And in doing so, we're increasing the rate at which we have voltage buildup. For the optimal solvent, we have fluorinated acetylnitrile. But what is really interesting here is that once you get past fluorinated acetylnitrile, further increase of this thermodynamic driving force actually leads to decrease in the rate at which voltage is being built up. In order to answer this, we go to the Marcus electron transfer theory, where for a donor to transfer its electron to acceptor, this rate of electron transfer is not only governed by the thermodynamic driving force shown here as delta G, which can be calculated very easily. In turn, the other factor that governs this electron transfer rate is called reorganization energy, which is the Gibbs free energy it takes for solvent molecules to reorganize themselves around the donor-acceptor pair in order to facilitate this electron transfer. So this KET, or electron transfer rate, is maximized when delta G, or your thermodynamic driving force, equalizes this reorganization energy. As you can see, the Marcus theory can beautifully explain our experimental data. Here, what we're saying is basically fluorinated acetylnitrile has the perfect LUMO level that gives rise to the perfect driving force that equalizes the reorganization energy in order for this electron transfer to occur. So this guiding principle of Marcus type electron transfer can explain not only our voltage tunability across all solvent that we have tested, but also it opens up the possibility for us to start tuning our donor substrate. In this case, what we're doing is by oxidizing our carbon nanotube lattice, we're lowering its Fermi energy, making it more and more easy for the electron transfer to occur, even for a single solvent acceptor, in this case, acetylnitrile. As you can see from the plot, increasing the oxygen doping level onto our carbon nanotube will actually increase the voltage production up to 700 millivolt for acetylnitrile. Higher oxidized carbon nanotube generates more voltage for acetylnitrile asymmetric chemical doping. We're also wondering about the power production performance of asymmetric chemical doping. So in order to test that, we do closed circuit current production measurement for asymmetric chemical doping. And we saw that as a function of decreasing device length, actually we get an increase in maximum power production. This may be odd at first glance, but it actually isn't, because our device has smaller and smaller internal resistance as the device length decrease. As a result, you generate more and more current given the same voltage. So this bodes really well for asymmetric chemical doping to be used as a microscopic power harvesting method. In doing the experiment for very, very small devices, we soon discovered that it becomes really cumbersome for us to drop asymmetrically these acetylnitrile molecule onto the carbon nanotube in a spatially asymmetric manner. So instead, what we do is that we take a layer of polymer protection layer, in this case PTFE, and coat our carbon nanotube in a Janus fashion, so making these Janus particles. In doing so, we can just immerse uniformly this Janus particle into a beaker of acetylnitrile, and that generates the electricity in order for us to drive energy harvesting. As you can see, current production is accompanied by uniform immersion of the particulated devices into a beaker of acetylnitrile. And our fabrication method is scalable and can be pre-patterned into different configuration for integration for colloidal electronic applications. So that concludes sort of the first section of this talk, which is the energy harvesting using photovoltaic-driven method. So the next question we ask is whether it is possible for us to turn that electricity we generate on board into a useful chemical signal. Biology uses hormones and neural signal transmitters for signal transduction purposes at the microscopic scale. These signals never really dissipate, and it can be amplified quite easily. So if we can achieve electrochemical conversion using onboard electricity, we not only gain the access of potentially communicating from one particle to the other, but also from an artificial cell to a biological cell. Okay, so just to recap what we want to do, right? We take these Janus particles fabricated before, and we immerse them into acetylnitrile, and the asymmetric chemical doping can produce electricity. And in this case, in this study, we want to turn that electricity into useful chemical conversion processes. Our model reaction for this is ferrocene oxidation, which occurs at the reduction potential of around 450 millivolt within our electrochemical window that is generated by asymmetric chemical doping of these Janus carbon particles. So the experiment was super easy. You basically put these particles into a beaker of acetylnitrile that has ferrocene in them, and we can just observe the color change as ferrocene is being electro-oxidized into ferrocenium. We can observe the reactivity using UV spectroscopy and measure quantitative yield for Janus particle that we can fabricate using the aforementioned method. The nice thing about an electrochemical reaction is that its reactivity is typically governed by reduction potentials. And with ferrocene, with different derivatives, we actually get our hands on a wide array of chemical substrate that has different reduction potential as compared to the parent ferrocene molecule. So here, what we do is that we take different particles and we can put them into a 96 well plate and just watch this reaction go as a function of time. If you plot the reactivity as a function of the reduction potential of all of these different ferrocene derivatives, we do see that we have higher reactivity for the ferrocene derivatives that has lower reduction potential in accordance to an electrochemical reaction mechanism. One thing I will mention is that we have two sets of data. Black ones are for the more oxidized single-walled carbon nanotube Janus particles, whereas the red ones are for less oxidized particles. As we know previously that more oxidized carbon nanotube generates higher open circuit voltage, hence having higher over potential to drive this electrochemical process, therefore achieving faster reaction rate. Knowing that our particle can drive electrochemical reaction in situ pretty successfully, we challenge our particle and ask ourselves whether it is possible for us to access organic electrochemical transformations inside acetonitrile. The reason being that most biological transmitters and signals are organic molecules, right? So here, as our model reaction for organic electrochemistry, we use partial oxidation of alcohol into ketone or aldehyde, mediated by this tempo, an electrochemical mediator that then couple themselves with our Janus carbon particle that provides the electricity needed for this turnover to happen. As you can see from this catalytic cycle, we basically chew up alcohol and then oxidize them into the aldehyde or a ketone that we want. This reaction occurs at basically quantitative yield for a large variety of substrate, showing excellent functional group versatility, which is quintessential for typical electrochemical transformations. In order to showcase the utility of having a particulate generator, we devised ourselves a selectivity optimization problem in which we try to oxidize the 1,2-propyl dialcohol. Basically, we can oxidize this alcohol in two locations, one at the primary location, generating this product, one at the secondary location, making this kind of product. In this problem, we ask ourselves whether we can basically screen for a condition that will selectively produce the secondary alcohol oxidation, which is the harder carbon to oxidize. And we tune our reactivity by changing catalyst loading and the Lewis acidity of the organic solvent. With our particle, though, we can do this screening using, again, a 96-well plate where we can just pipette different solvent conditions into the wells that has our particles in them. And in one fell swoop, we can basically identify the condition at which we can generate 90% yield and essentially 100% selectivity towards the secondary alcohol product. The particulate form factor of these particle generators couples actually very nice into NMR experiment for us to track the kinetics of our oxidation reaction within an NMR tube in real time. So as you can see here, the particles are at the bottom of the NMR tube and they do not block the magnetic wavelength for NMR study. And we can basically track the growth of our product and the disappearance of our reactant within an NMR for this electrochemical process. So I think, hopefully at this point I convinced you that we have the ability to convert solvent energy into electricity and use that electricity for electrochemical processes that can drive the synthesis of signaling molecules that can later on be used for communication purposes. But to complete the loop, we need to be able to make electronic circuitries on top of a colloidal particle in order to pick up the signal, record the signal electronically, and maybe even process it digitally. Right, so this is going to be the next topic of focus. In order to achieve that, we utilize a class of materials known as two-dimensional materials. So these are atomically thin materials such as graphene, MOS2, or hexagonal boron nitride. Just like carbon nanotubes, these materials also have tunable electronic properties through the composition change of this atomic lattice. Within the material, we have free access over a huge variety of electronic elements, for example, conductors, semiconductors, and insulators within this atomically thin lattice. And the atomically thin lattice allows us to flexibly fold these two-dimensional materials onto any surface that you want, which is another attractive characteristics of two-dimensional materials. So here comes the idea. You take a colloidal particle, treat the 2D material as origami papers. So you can pre-pattern electronic circuitry within the two-dimensional materials with different composition. And then we fold these two-dimensional material around a dumb colloidal particle in the middle, making these microscopic dumplings that has two-dimensional material covering its surfaces. These dumplings, or raviolis, depending on where in the world you come from, basically have an electronic skin and a polymeric support in the middle. The key question here, though, is that how do we fold the 2D material around the colloidal particle and use a molecular scissor to cut the 2D material along these edges to make these microscopic dumplings? And it turns out the answer to that is controlled fracturing. I know some of you might be surprised to hear that fracturation of a material can be actually a controlled process. But indeed, you can actively control the fracture pattern of a material, actually a problem that was solved almost exactly 100 years ago by Alan Griffith. So as we fracture a material and advancing a crack within that given material, if you plot the overall system energy as a function of the crack length, it basically adopts itself this concave down shape. And you have the vertex of this energy landscape at the location of a critical length scale that is called Griffith length. So if your seed crack within a given material is shorter than the Griffith length, you would notice that increasing the crack length is actually not advantageous because that increases overall system energy. On the other hand, if your crack is on the right-hand side of the Griffith length, you basically say the crack will propagate forever and the material will fracture. So Griffith length really controls the fracturability of a given material. Griffith then went on to calculate the algebraic dependence of Griffith length as a function of a variety of different material parameters. And specifically, he had noticed that the Griffith length is inversely correlated with the strain of a given material. So that is very important, meaning increasing the strain will correspond to a lowering of the Griffith length, making the material easier to break. Here comes the question, whether it is possible for us to apply Griffith theory into two-dimensional materials and pre-pattern a strain potential within the two-dimensional material and cut the material around the edges, make this controlled fracture propagation a fabrication method for colloidal electronic devices. We call this method automatic perforation of two-dimensional materials, or autoperforation in short. To do this, you take one layer of two-dimensional material and inkjet print an array of micro-printed polymeric pillar. And then you take another layer of 2D material and sandwich this entire array, getting essentially a sandwiched structure that has either graphene, MOS2, or other types of two-dimensional material that you still want. If you imagine now what is happening within the given set of 2D material that is folding over into this array of polymeric pillars that we have printed, if we zoom into one of these particles, you see that as the 2D material folds over the circularly shaped pattern, very analogous to the process in which you fold a tablecloth over a circularly shaped table, right? Naturally, you will gain higher strain on the edge of this table, and translating to our molecular picture here, we're getting more and more strain around the polymeric pillar. So according to Griffith's theory, a randomly generated crack will get arrested by this ring of maximum hoop strain, and it will actually cut along the edge of this polymeric pillar that we define using our inkjet printing. And experimentally, this happens basically exactly as what we have predicted. These particles will get cutted around the edges, and the 2D material will only survive at locations where it encapsulates the particle. And as a result, when the particle gets lifted off, it has a perfect encapsulation by two layers of 2D material, one on top, one on bottom, and it can travel around as a colloidal particle. So I want to introduce how we can then build chemical sensing and information recording to this kind of electronic colloidal particle that we have just built. In terms of chemical sensing, we leverage something called a chemical resistor that is built within the two-dimensional material themselves, which changes its conductivity based on the chemical analyte it detects. So we're making these microscopic dumplings that has the two-dimensional material skin, in this case graphene, and the polymeric interior. By grafting aiming functionality onto the graphene, we're able to use the aiming functionality to grab onto gold nanoparticles that are distributed into a groundwater source. So by harvesting our microparticles back and basically query its surface conductivity, we can measure whether or not our particle has seen gold nanoparticles in groundwater. Alternatively, you could functionalize the graphene surface with something called an NTA ligand, or nitrile triacetic acid ligand, and then use it as a zinc cation sensor, because NTA ligand chelates onto zinc cations, and these particles can be injected into a soil matrix, and after they travel from the soil matrix, we can sort of harvest these microparticles and again query them for surface electrical conductivity. Long story short, we can get a very selective and sensitive sensor to turn chemical signals into electrical conductivity measurements. What about memory, digital memory? In terms of building digital memory into our colloidal particle that are encapsulated by two layers of two-dimensional material, one on top and one on bottom, we leverage the fact that we have a vertical dimension in our electronic circuitry. From top to bottom, we have a two-terminal device. And by printing memory-resistive black phosphorus inside, we can make these vertical memory-resistive pathways that makes our particle able to record digital information. So as you can see here from the current voltage trace, we can write in an electronic memory by turning this vertical conductivity from its off state or non-conductive state to on or conductive state by driving a positive voltage across, and we can switch it off by driving a negative potential across it. So this then renders our particle into sort of multiple random access memory devices that are also reversible. So here, as a demonstration, we showed that we can write the letters MIP within a single particle through vertical conductivity map. So here we're using an electrical probe to selectively write in the letters, and we're also reading out the letters and erasing the letters and write again. So this is an entirely reversible process for us to store digital information within a cell-sized particle. So if you think about what we have done here, we take a colloidal particle and then wrap 2D material around them using controlled defect assembly to render a fabrication method for these colloidal electronic devices. I don't have time to get into this study, but in this beautiful work, Vladimir Coleman in our lab were able to integrate chemical sensor and a memory-resistive device and onboard energy harvesting solar cell makes this particle into a little computer that can do logic computation on board of a single particle. So in a very realistic way, by merging two-dimensional electronic materials onto the mobility that comes with the colloidal particle, we can then extend artificial electronic devices into previously inaccessible locations. I think at this point, we'll take a step back and ask ourselves that these particles, while pretty powerful, are nowhere close to our computers in terms of the ability to do computations, right? They cannot run Microsoft Word, they cannot run MATLAB. So are there any ways for us to think about colloidal electronic system such that when they work with each other, they will be able to access cooperative behavior between these less powerful individuals and access a complex function that is only available at the system level? This kind of emergent behavior is prevalent in nature, as you can see from this photo. But there's one critical missing piece in our puzzle for colloidal electronics in order to access cooperative behavior, and that is locomotion, controlled active locomotion. In order to build locomotion as a functional module into our colloidal electronic devices, we changed our autoperforation fabrication technology and devised an asymmetric coating of metal, any metal that you want, onto our graphene layer, which is the two-dimensional material that coats the particle. As a result, this was able to give us, after autoperforation of the two-dimensional material, a cell-sized particle that are not only encapsulated by graphene from top and bottom, but also has this asymmetric layer of platinum coated onto the graphene material. Now, this simple addition of the platinum patch actually introduced a useful function. In this case, we're building a locomotion module to our particle because platinum is known to decompose hydrogen peroxide, making these oxygen bubbles. And then we can use the bubble as a propulsion method to propel our particles forward. Finally, we have access to locomotion. And so I'll introduce to you how then we can use the fact that we have this primitive way of locomotion and access systems level behavior for dynamic device assembly. We take our little particle that has a platinum patch on one side of it, and then we turn the particle sideways, arrest it at the interface between air and the liquid phase, with the platinum facing downward, pointing at the hydrogen peroxide solution. Now we're still making a bubble because hydrogen peroxide gets decomposed into oxygen in the presence of platinum, but because the particle is sitting on top, it traps the bubble underneath, and the bubble becomes larger and larger, as you can see from these photographs, but it never really gets popped for a very simple reason, because as the bubble grows larger, it blocks the platinum patch's access to more hydrogen peroxide. So this is a self-inhibiting reaction. I should tell you one additional information here. As particles are arrested at the air-water interface, it's subjected to a capillary interaction that is broadly known as Cheerios attraction. For the very reason that it's responsible for the fact Cheerios chip will always come together in your breakfast milk bowl. So this Cheerios attraction between these two particles can serve as an interparticle attractive force that will drive these particles together. So while that we have mentioned that this one single particle, as it's arrested at the air-water interface, it doesn't do much, right? It reaches basically an equilibrium phase where the bubble production rate dramatically slows down. I'm gonna play this real-time video of one single particle as I go through this slide. But note what happens if I put two exactly same particles together at this air-water interface. The bubbles gets attracted by each other through the Cheerios interaction we talked about before. And as the bubbles get closer to each other, they will merge into a single bubble in the middle, because by merging into a single bubble, it decreases surface area and has its surface energy. As the bubble collapses into one bigger bubble in the middle, now the platinum patches can see more hydrogen peroxide again. So it continuously generates the oxygen that fills into the central bubble, and eventually the bubble pops. And this bursting of the bubble will eject the two particles away from each other, forming this mechanical oscillator. So this is quite surprising, because there's nothing in our system that has prescribed a beating or an oscillation that has a given frequency. But indeed, if you track the particle trajectory, map out what we define as the breathing radius, which is basically another way of saying the distance between these two particles as a function of time, you can see that this oscillation happens at a very defined frequency. And this frequency can be further controlled by the concentration of the chemical fuel you have within the system, in this case, hydrogen peroxide. Because as the hydrogen peroxide gets more and more dilute, the oxygen generation rate is slower and slower. You can see that as we decrease hydrogen peroxide, we lower the frequency of such oscillation. And we can understand what is going on analytically using a very deterministic simulation model we have created, which qualitatively and quantitatively agrees our experimental measurements. At this point, you might wonder what would happen for a multiple particle system. And because this is ongoing work, I will cut a long story short and just tell you that if you have more particles in the system, you get access to even more complex and interesting behavior. So for a 5-particle system, we still exhibit in our system this very defined oscillation frequency. And this again agrees quantitatively with our simulation. And it's nice to have a simulation program here, because the simulation can allow us to access conditions at which we cannot perform an experiment. For example, what would happen if you have 50 particles in the system? What would happen if you have 500 or even 5,000 particles in the system? So at this point, I just want to take a step back and appreciate what has been achieved here with these very, very simple particles. Controlled and synchronized oscillation is a fundamental aspect of life. You can see in these microscopic organisms, these kind of contractile and expansion behavior at a given frequency is fundamental to how they go and search for food. And in these slime molds, single-celled organisms, they work with each other and doing a global contraction and compression. This is fundamentally how they move across a distance. Synchronized oscillation is not only a fundamental aspect of microscopic organisms, it's actually also a fundamental aspect of life found in us, in humans. So seen here are cardiomyocytes that, when oscillated together, forms our heartbeat. So it's absolutely critical for these single cells to sort of do their mechanical oscillation or compression expansion in a very defined frequency. Otherwise, we would suffer heart failure. These are very complex biological networks and very complicated behavior that was basically realized using a platinum patch underneath a particle at the interface between air and water. I think that highlights the potential for colloidal electronic devices to be used for even more complex things in the future. So in my mind, we cannot help but wonder what kind of magical function could occur in our colloidal electronic systems if we can actually integrate all of these functions that I mentioned today. So with that, I want to thank the people that has contributed to the talk you have heard today. Yuichiro, Jingfan, and Pingwei were my primary collaborators within the group. And of course, I acknowledge the help and guidance that I got from the entire Strano research group. I do want to mention several individuals. First and foremost, Michael, my thesis advisor. These mantras that he has definitely carried with me in my future academic life. And I want to acknowledge Will and Kartish, who are my primary committee members. Both Will and Kartish have greatly exceeded what was written on the job description of PhD committee member. I look forward to continuing this mentor-mentee relationship with you. Some of my best moments spent at MIT were inside actually a classroom teaching. If you want to be the best, you have to learn from the best. I'm really fortunate to have P8 for transport phenomena taught by Bill and Martin, and chemical reaction engineering taught by Michael and Klaus. I want to mention the presence of my collaborators. Their advice and guidance have shaped in many way, shape, and forms how this thesis project has turned out to be. I also want to acknowledge undergraduate students that I have worked with over the past 6 years. I'm super happy to see their growth every step along the way. I want to especially highlight the presence of Jack, Greg, John, Stacy, continuous source of inspiration, MIT Rowing Club, the Dragon Boat team. Some of my best memories during PhD were actually spent on the Charles River. So many friends that I want to acknowledge, and it's impossible to fit all of you onto a single slide. I'm very fortunate to have grown up in a very, very happy family. And my parents are actually my biggest role models in life. A constant source of inspiration. Without you, none of what I have presented today would be possible, which is okay because without you, none of this would have mattered. I want to thank my best friend and life partner, Stacy. Stacy and I met here at MIT when she was doing her MBA. 4 years later, she has acquired 2 degrees and I've got nothing to show for it. You believed in me even at moments when I do not believe myself. I definitely share half of the credit of my PhD work with you. With that being said, if I do not pass this PhD defense, I will blame at least 50% of that to you as well. All jokes aside though, I want to thank you all for coming to the PhD defense. It has been incredible. I really appreciate your presence. If you are still awake, I will be happy to answer any questions that you might have. With that, I will yield the floor back to the thesis presider. Take it away, Will.
Thank you, Albert. Feel free to unmute yourself and clap.